Hi, everybody. My name is Jivesh Ramduny, and I am currently a third-year PhD student at Trinity College Dublin. Today, we are focusing on the workshop, which is about an introduction to methods for analyzing large neuroimaging datasets. And we'll be presenting a number of different topics, and these topics are actually part of an online book that we have created and will be uploaded very soon on the OSF platform. And the book is about methods for analyzing large-scale datasets. So when we talk about reproducible neuroimaging analyses, we need to be able to understand what reproducibility means, because there are a number of definitions that have been associated with reproducibility. And also it is important for us to understand, how do we do reproducible neuroimaging analysis? So, firstly, let's look into the definition of reproducibility. So the term reproducibility is basically being able to obtain the same results as a prior study using procedures that are closely matched to those described in the original research. So why do we care about reproducible neuroimaging analyses? So if you may be familiar with the reproducibility and replication crisis in research, you may be aware that it is incredibly difficult to replicate or reproduce someone else's study. So following the crisis in research, which doing reproducible neuroimaging analyses has become a cornerstone for neuroimaging research. And to get started with understanding how we can proceed to do reproducible neuroimaging analyses, it is important to start off with data sharing. So data sharing is important not only for us, but it is also very important for labs to be able to gather a large sample size so that the results that they get are less likely to be inflated or less likely to happen purely based on chance. So data sharing has not become only important, but it is expected now that people should follow this norm in neuroimaging. Equally, by sharing your data, now there is a strong emphasis to share your codes that you have written to do your analyses, because at the end of the day, we are doing science, and doing science is not doing miracles. And sorry. And while there is a tendency now to move towards data sharing or pooling data from multiple sites, we are also sharing our codes, and this allows you to replicate or reproduce analyses in high impact journals, but also based on the essence that you are able to gather thousands of participants to boost your sample size. So we are providing a very comprehensible practical description in this workshop, but also as part of our book chapter, which will be made available very soon. So when we share data, there are specific norms that one should follow. So today, openly available data sets follow what we call the FAIR principles. So the FAIR principles are basically a set of characteristics of data and digital objects to maximize their reuse by the scientific community. So here, these principles do not only focus on maximizing data reuse sharing, but also to be able to deposit such data so that other people in the world can access and download these data. So we don't want things to be messy. We want everybody to follow a specific standard so that data sharing can be made very easy. So what is this FAIR principle? So the FAIR principle basically requires your data to be first findable, meaning that you have sufficient rich metadata and having a unique PID, so which is a unique identifier. Second, you need your data to be accessible. So you need to provide your data and the metadata uh, via trusted repositories such as GitHub or OpenEuro. Third, you want your data to be interoperable, such that your data and the metadata will use a specific formal and shared format that is agreeable by the neuroimaging community, and such example is the bits format. And lastly, you want your data to be reusable. And what do I mean by reusable? It means that the data should be described with clear and understandable attributes. And also there should be a consensus about the license for you to reuse these data. So, like I said before, data sharing has been gaining a significant traction in the last couple of years. And there has also been a development into hosting these large scale data sets. So such a platform for hosting open source data is called OpenNeuro. So you may have heard about OpenNeuro. So if you are a PhD student wanting to use large scale data sets, OpenNeuro is a very good place to start with. So OpenNeuro has about 604 datasets, and they are derived from mostly MRI modalities, but you also have EEG data or PET data. So it's not restricted to only MRI modalities. So if you look at the 604 datasets, you are able to see that there are about 501 datasets which will have structural MRI data, and then you have about 53 datasets that will have diffusion data, and then about 445 datasets which will be having functional data. So if you want to have a go at a specific data set based on your research question or your research project, you have a wide range of choice to select from. And if you look at the top right, the graph, you can see that every year there is an increase into the number of data sets that are hosted on OpenEuro. And as such, you can see that now, as of now, OpenEuro has crossed over 20,000 participants. So these kind of sample size would be impossible to get with individual studies or with you collecting data in a single lab. So this shows the power of data sharing or pooling data from multiple sites. So apart from OpenEuro, we also have a broad spectrum of open access datasets. And these datasets, as you can see here, would be probably the most commonly used one, or probably the ones that you would be most familiar with. So if you look at the list of datasets, and this is adapted from the Madden paper, which provides a beautiful overview of the open access large datasets that exist currently. So for example, if you look at the UK Biobank, they have crossed over 50,000 participants across the lifespan. Then you have the Healthy Brain Network, which targets mostly adolescents and neurodevelopmental population. You have the Human Connectome Project. You also have datasets that not only uses structural and resting state or task-based fMRI, but also using movie watching fMRI. So like the naturalistic neuroimaging datasets. You also have one of the largest adolescent datasets, which is the ABCD dataset, crossing over 10,000 participants. And you can see that you have huge resources that you can leverage and use and apply to your own study. So the question is, then how do you access these data? Because these data are publicly available, but how do you use them? And here you would be, as a PhD student, you would be familiar with these open access datasets, and these datasets are commonly used on a daily basis. And here you also have datasets that not only focuses on human neuroscience, but also focuses on the primate neuroscience. So you do have a large um, option that you can select from. So here, I am going to focus on one example. So I'm going to show you how we can access a specific data set on OpenNeuro. So OpenNeuro, like I said before, it is an open source data hosting platform. And there are different ways that you can follow to download data sets. So here, one of the ways that we're going to use is called data-led, but you can also use the Amazon server to retrieve these data. So here we're going to look at how we can download the Aomic dataset via data-led, which is hosted on OpenNeuro. So to be able to access data from OpenNeuro, what we're going to do is we are going to use Jupyter Notebook. So Jupyter Notebook, I'm going to talk about this more in a moment. But here, what we're going to do is we are going to use the data-led. And so here, instead of downloading hundreds of gigabyte worth of data, we are going to have a smart way to access these data. So first of all, what you need to do is you will need to download data-led or install data-led, because it is not included as part of the Jupyter Notebook. So once you have installed data-led, then you will need to download a dependency called Git Annex. And Git Annex is basically a Git for data management. Okay, so here is an example how you can download and install data-led. So you will need to have pip install data-led, okay, because it is not included in Jupyter Notebook. And after you would install data-led, then you need to install Git Annex, okay? And Git Annex uses a package manager called Homebrew. And with Homebrew, sometimes it is already installed, sometimes it is not. So for example, if you are using a macOS, you can copy this line of code into your terminal, paste it, and then it will install Homebrew for you. So once Homebrew is installed, you can download the Git Annex, which is a Git for data management using these lines of codes. So once you run these lines of codes, it will ask you for your password. So the password that you use to access your laptop or your computer. Then once you enter it, it will download Git Annex for you. So after downloading data-led, then we need to use it. So here, the Aomic dataset is the dataset that we are using throughout this workshop. And if you look at the size of it, it is about 407 gigabytes. So it makes it almost impossible for everybody to download it, but also it is not good practice for different people in the same lab to download things locally, because it is just a waste of resources. So instead, what we need to do is, what we can do is we can clone this dataset, which is on OpenNeuro, and we just need to provide the directory of the dataset that we want. And this way of doing things means that we use data-led, and then we provide the symbolic links, which is the location of where the actual dataset is hosted. So here you would use a data-led install, and then you would provide the URL where the actual data is. And If you do run this line of code, it will take a few seconds. It will create a folder for you called ds002790, and then this will be available in your downloads directory. So here, once you have this folder in your downloads directory, then you can have a look into it and see what's going on. But here, what you would be able to see if you check the size of the data frame, of the data set, you will see that the size will be just KB or kilobytes, meaning that you don't actually download the data. You are downloading the metadata where the actual data would be. But let's say you do want to use the fMRI data for a specific participant or for all the participants. So this data set has 226 participants. What you can do is you can actually get the actual data, the actual image, and load it into your notebook. Again, you're going to load the actual image, but you're not going to download it into your computer, because imagine if you have to download over 400 gig of data, this would be too much. So here, a smart way of doing things is you get the data from data-led. So here we use data-led get, okay? And then you will provide the file that you want to access. So here, the file is in this directory. So I have downloaded it. So it should be in my downloads folder under the name ds002790, which is here. And then I will be using the already preprocessed data. So here, the good thing is some datasets are already preprocessed by fMRI prep. So here, the preprocessed data is under the directory derivatives. And then I will specify the entire path to the image that I want. So if you load this, then this is about 193 megabytes. Again, you are not downloading it specifically to your computer. You're just loading it into this notebook. And you can do this for all the participants in the dataset without having to download it. Then you may want to check whether the data is actually correct, or if the way that you have done this is actually correct. So then you can view this image. So here you can view this image using Nylon, but again, you need to do something. So the fMRI data is usually a 4D image, which means that the fourth dimension, it means that it is the time direction. So what you need to do is you need to average this 4D image across the fourth dimension of time, so that you get an image which is in a 3D format. So luckily, we can do this using Nylon. So you would import Nylon, the package, and then there is a way to do the average across the fourth dimension. So you would provide the mean underscore img, and then you provide the path to where you downloaded your fMRI data, and then you can view this using view underscore img. Okay. And in this way, you are able to visualize your 3D image of that participant after you have accessed it. Okay. So again, this is one basic way using data-led of how you can access the fMRI data of a specific participant. So let's say you want to access more than one participant. All you have to do is you can just add lines, and then you will load all the participants that you want in this notebook. Okay, so let's continue for this workshop, and then you'll get to know more about what is a Jupyter notebook. So like I showed you, here you have now you kind of know how to use data-led to get a specific data set on Open Neuro. Hopefully this is a bit clear now, or how to use Datalet. But it is very important to understand how you can write your codes in a reproducible format. So the most popular way, or the best way to write reproducible codes, is to use Python notebooks. Now the question is, why do we use Python? Why Python has become so popular? This is because Python is free, so you don't need to pay for it. And it is a very popular open source programming language, not only in neuroimaging, but in other disciplines. So because we tend to use Python increasingly now, people have started using the Python notebooks. So just now I showed you how to download a specific data set using OpenNeuro with Jupyter Notebook. So this is what we mean by Python notebook. So it is an easy way to write reproducible and portable code. So like an actual notebook or diary, it makes use of Markdown. So Markdowns is basically a markup language that allows easy plain text formatting. So you are able to describe your project, your codes in words, and then you add the lines of codes, and then you can have some formatting, some headings, comments, hyperlinks. You can make the text bold, italics, underline even. So it works really like a notebook. But the Python notebook, or sorry, I should say the notebooks uh, themselves, they are not restricted to Python. It is also commonly used in another open source program, mostly with statistical analyses. And this is what we call the R Markdown. So if you have used R before, you would know that R Markdown is essentially what Jupyter Notebook is. So they are very similar, but Jupyter Notebook is more scalable and more appropriate for neuroimaging analysis. So then, so then how do you install Python? So we know now that Python is very useful. The notebooks are very useful. So you can install Python on your computer. So that's what we call to install Python locally. Or you can use Anaconda. So Anaconda is a suite of tools, mostly for data science, but you get different versions of Python, even R, and then you can choose the different notebooks that you wish to run. So you have Jupyter, you have Jupyter Lab, and it is very, very good. So Anaconda is one way of installing Python. The second way is you can just download Python on its own. And lastly, you can also run the notebooks, not from Anaconda, but something called Google Colab. So Google Collaboratory is a web-based platform which is more carbon neutral, which uses less energy to perform similar um, similar computations as with Jupyter Notebook using Anaconda. So now you know that you can use Python and Python notebooks to perform reproducible neuroimaging analysis. So now you need to get started on coding. So I know that the hardest thing to do is to get started, but there are plenty of resources online. So if Python has a wiki, or you can also use other learning platforms like Coursera, ETX, and Udemy to help you in your programming journey. And they have courses from all kinds of institutes, but also these courses target a wide range of audience, from beginners to experts. So it will require time and practice to know how to code in Python, but you need to get started. So once you get started, I will strongly emphasize that you guys spend time on learning some good coding practices, because at the end of the day, you want to share your code, you want to share your analysis to the neuroimaging community. So you want your codes to be well structured, clear, and concise. You don't want your codes to be complex or to have unnecessary repetition or to be what we call spaghetti code. So one way to make your codes very structured and clear is to include comments. So comments is a way to explain the purpose of your analysis of a specific function or a line of code by starting the line with the hash symbol so that you would basically explain what your code is doing. And also for your future self, you will remember what you did a year from now, or even when you would share your codes or your notebooks to the neuroimaging community. This would be highly beneficial to an external reader. So according to Kirsty Whitaker from the Alan Turing Institute, comments should represent about 40% of your codes. So please do invest time in learning how to code. Also, it is also beneficial to know how to debug your codes. So debugging means that trying to figure out why your code is not working or producing the wrong output. But this is a skill that you would develop over time, and it involves a lot of trial and error, meaning that you will try something, doesn't work, you could crash this, you try something else, or even you use Google. No one, even experts, do not know everything about programming. They would Google. So, Google is your friend, use it, or just try and try to solve it. If it crashes, try again. And surely someone on the internet has already solved the error. So let's look at the notebook again to be able to understand what would be an example of a good and bad coding practice. So this is another notebook that we are going to share, and this will form part of the book chapter that I mentioned before, and this book chapter will be part of the book on methods to analyze large neuroimaging datasets. So I want to show you, so this is an example of another notebook. So here you can see that here I have my markdown descriptions of the code is doing. And here you can see that in your imaging, you will import some packages that will allow you to do some computations. But you can see here that I have included comments, so everything following the hash symbol that tells me and tells you what I am doing, what packages I'm doing, I'm importing. And here I'm loading a file. It tells you what file I'm loading, what does it do, what size the file is, and so forth. So it's very important to include comments in your code because it will help you later on. And secondly, as a good programming practice, the name of your variables are very important. So here you can see that I'm using behave underscore df, which shows that I'm loading behavioral data. And people tend to use lowercase, uppercase, or underscore, and even a hybrid, so a mixture of, lowercase and uppercase. So it depends on your preference, which one you like, but you need to be consistent. So so here you can see that, but I am naming the same file using different variable names. So here behave, and here behave underscore df, and here behave underscore hbn underscore data. But also, let's look at a bad naming convention. So here you have temp. So people use temp for temporary computation. So if you have temp, you don't really know what temp is doing. Or if you have two temps, you don't know what they are, right? And also people tend to use names like final variable, final something. So you don't really know what final is again. So it's a bit confusing. Or sometimes you do have very long undescriptive names, which basically is very complicated. So try to keep it short and intuitive. And lastly, if you use final underscore temp, Again, confusing, it's undescriptive, and you don't know what's going on. Also, it includes a number. But you need to be careful though. If you just use a number to name your variable, it will not work because Python will interpret this as a number. Okay, and then you can also print the shape of a specific variable. So this helps you to check your codes, whether the codes are giving the right or wrong output. And this is what we call doing a sanity check in programming. And you should also be careful of spaces, of white space, because sometimes here you can see that it's fine because I have, here you can see that I have additional spaces. So I have two spaces here, and this is what we call the white space. It's very hard to identify that kind of error if it happens because it's what we call the unseen error. And here, including one space is enough. And similarly, you can see that there is a space here, and here you have two spaces, so you should be very careful. Okay. And lastly, we talk about indentation. So indentation is basically when you have a for loop. So if you're trying to do a computation in number of times, let's say reading, manipulating files for every participant, you use a for loop. So for every participant, you do something. So you tend to indent your code, meaning that you have a space or a tab so that Python knows you are doing indentation. But the problem is sometimes you have too many spaces. So for example, here, you can see that I have my for loop, and following the colon, Python will already bring it for you to start. But sometimes like you may have a mistake where you have no spaces at all. This will crash, will reset an error. And here you can see that you have too many spaces, which is not a good practice. Okay. So these are some basic good and bad coding practices. So hopefully this will be useful for you. So I have showed you what a Jupyter notebook looks like. So you have Markdown, you have cells that correspond to descriptions, cells that correspond to codes, and this is an aesthetic way for you to conduct your analysis in a reproducible way. And then you can share it with your supervisor, colleagues, and the neuroimaging community. So before I finish, I wanted to talk a bit more about some neuroimaging packages. So Python is very powerful. So you have a wide range of packages to help you perform a wide range of analyses. So univariate analysis, multivariate analysis. Some of them are more specific to machine learning, but overall, these are the most commonly used packages that you will experience. So NumPy, Pandas, Stats Models, Scikit-learn, Nylon, NL Tools, Penguin, Matplotlib, and Seaborn. So here we are presenting some of the most commonly used packages, but of course, depending on your research, you will be using and experiencing many more. So this is just a way to show you about the most commonly used packages and help you to be familiar with and to get started. So this is useful, especially when you're learning how to use Jupyter Notebook. So thank you very much for your attention. And I hope to see you in person. If not, I hope you enjoyed the virtual workshop. And if you have any questions, you can reach out to me by email or you can follow me on Twitter. Thank you very much.